What is the influence of HLA genotypes on coronavirus infection? This is what we will discover in this presentation. Are you interested? So come with me. Hello guys, welcome to the HLA channel. My name is Gustavo Milson. I am a PhD in immunogenetics and I have been working in an HLA lab for more than 14 years. Now I work for a company that sells the best HLA kit by NGS in the world. Yes, I'm the sales guy too. Well, I love to study HLA and I love to do these kinds of videos. I love to make videos. So I'm here to study a little bit more, to share with you what I'm studying and to practice English. As you can see, I'm trying to improve my English. So this is a great opportunity for me to practice my English while I'm studying something I like, I enjoy to do. Do you think HLA is associated with SARS-CoV-2 infection? Stay with me and let's discover it. Well, we're now in a global pandemic caused by SARS-CoV-2 virus. COVID-19 is caused by an acute respiratory syndrome related to coronavirus 2. The SARS-CoV-2. Of course, you are living it now where you live. This is a mortal infection. For example, in China, we have 5% of mortality. Some parts of the world, we have 15% of mortality. People with chronic illness, for example, in the lung, kidney, liver, cardiovascular disease, and diabetes, and with compromised immune system are at high risk for serious illness. And of course, molecular markers can be very effective in identifying susceptible and resistant individuals to SARS-CoV-2. HLA tests are cheap, are fast, and why not use HLA tests to identify people with a higher risk of COVID-19 infection? We know many viruses, many bacterial infections that are associated to the host genetic. So why not study HLA, genotype, and COVID-19? So if you can understand a little bit more the host genetic, so we can fight more efficiently this virus. It is quite evident that several factors contribute to determine susceptibility to SARS-CoV-2 infections and the severity of COVID-19. In this figure proposed by Pogero, we observed several factors that have been studied as aspects that can influence the progression of the disease. Lifestyle, in terms of habits, nutritional quality, physical activity, drug use and smoking, and socioeconomic parameters, access to health system, medication levels, and everything. So lifestyle, environment, comorbidities, viral genetics variants, immunogenetics, host genetic variants. So everything may contribute to severity of the disease. If we have many things that can influence in disease, why study HLA? In this paper, we have a beautiful explanation and I will try to read for you. Individual genetic variation can help explain the difference in immune response to a virus in a population. In particular, understanding how the variation in HLA can affect the course of COVID-19 can help to identify individuals at greater risk of contracting the disease. HLA typing can be quick and inexpensive. Correlating the HLA typing with the COVID-19 test, when feasible, can improve the assessment of the severity of viral disease in the population. After development of a vaccine against SARS-CoV-2, individuals with high-risk HLA types could be prioritized for vaccination. Good. So this is why I study HLA, because it can help us in the COVID-19. HLA encodes cell membrane proteins responsible for regulation of the immune system. We have HLA of class 1, HLA A, B, and C, and these HLA present peptides from within the cell on the cell surface. If the cell is infected with a virus, peptides of viral origins will be presented so that the cell can be lysed by CD8 positive cytotoxic lymphocytes. On the other hand, we have class 2 molecules, DRB1, 3, 4, and 5, DQ alpha, DQ beta, DP alpha, and DP beta. These class 2 molecules have exogenous peptides which have been captured outside of the cell for CD4 positive lymphocytes, which in turn stimulate B lymphocytes to produce antibodies against that specific antigens. So the polymorphism of class 1 and 2 molecules affects the peptide binding groove, since it varies the amino acid sequences that can be housed within the groove. So these peptides are presented to the T cell CD8 positive, for example. In this example, okay, we have a beautiful class 1 molecule that I draw, okay, and now we have a CD8 positive cell attached to this HLA molecule. If you have variation, modification, mutation in this groove of the HLA molecule, it will affect the peptide will be presented. Okay, so if you have mutation in HLA molecule, different peptides will be showed in the groove, right? Therefore, HLA genes are crucial in MHC peptide interactions and in differential susceptibilities to viral infection. Many studies have associated HLA to SARS-CoV-1 in the past. So, for example, allele at risk allele, HLA alleles was found, for example, HLA-B 4601, HLA-B 703, CH01, and DRB1 1202. On the other hand, protection HLA alleles will have DRB1 3301 and C1502. So let's talk about some HLA papers. First of all, I would like to talk about a paper that it is not associating HLA with SARS-CoV-2, but to another disease. For example, Tian, in a genome-wide association, she found many diseases associated with HLA. For example, chickenpox, herpes zoster, cold sores, mononucleosis, all caused by members of human herpesvirus. They are associated to class 1 because they are virus, right? On the other hand, we have disease associated to class 2, HLA class 2. For example, bacterial disease, especially tuberculosis, scarlet fever, and childhood ear infection. All of them are related to class 2 HLA molecules. In this paper, Anastasopoulou presented a summary of reported association between human genes and differing severity of COVID-19. We have here a lot of genetic factors related to COVID-19, and we can see the B4601 and B1503 conferring cross-protective T-cell based immunity. It is possible to suggest biomarkers that predict the development of severe COVID-19. Again, we can see that B4601 and A2501 serving as biomarkers to COVID infection. In the same paper from Malkova, we can see this association through different populations. Okay, so we can see how these markers can be used in different populations. Returning to the famous study by Nguyen, they performed in silico analysis of the binding affinity of MHC class 1 molecules with viral peptide with 145 HLA A, B, and C genotypes for all SARS-CoV-2 peptides. They looked to the overall population in the world, and then they observed where there were more frequency of a particular HLA or less frequency. So what they found was that HLA B4601 allele could increase susceptibility to COVID-19 since this allele had fewer bind peptides predicted to SARS-CoV-2, and in contrast, HLA B1503 can provide protective immunity based on T cells. In general, at haplotype level, they stated that HLA A202, B1503, and C1203 exhibit the highest, and HLA A2501, B4601, and C102 exhibit the lowest predictive repertoire. This is a difficult word, isn't it? So the lowest predictive repertoire of SARS-CoV-2 epitopes. Oops. Well, Pisant performed a geographical epidemiological analysis to find if in an Italian population there are a particular frequent haplotype and HLA alleles, right? So they found that this haplotype had a regional distribution overlapping that of COVID-19 and showed respectively a positive suggestive of susceptibility and negative suggestive of protection, significant correlation with both COVID-19 incidence and mortality. Oops. One of the most interesting finds that emerged from the analysis of this population in Sardinia was the total absence of this haplotype. Okay, this haplotype is the third in order of frequency in Sardinia. So we can say that this haplotype can provide protection against SARS-CoV-2. Tomita and colleagues propose a hypothesis that HLA genotypes may impact the difference in morbidity and mortality of COVID-19 across countries. And what they found was a possible association between HLA A201 and an increased risk of COVID-19 due to lower capacity to present SARS-CoV-2 antigens. Wang in China performed an NGS study with the 11 loci, and if he performed an NGS, so we can expect a high resolution results. And what he found was that the HLA C729 and HLA B1527 alleles were higher in patients with COVID-19 than in the control population. This is an interesting paper for me. Ben Liamani showed that in monocytes, he showed less HLA-DR expression in circulation monocytes, which created immunodepressed conditions for the host response. Okay, so if you have less your expression in your monocytes, so you get worse in the COVID disease. Again, we have many factors that may influence COVID-19, and we have always HLA genes involved, or in the risk level or in the protection level, okay? But we always have HLA involved, as we can see again in this paper from Litera. To summarize, I included here all the risk alleles and protection alleles of HLA. And to finalize, I'd like to suggest this study, and mainly because it is a Brazilian group, okay? But it is a very nice paper, very recent paper, where they made a systematic review of candidates gene association study. Okay, so this is my suggestion for you as a very, very recent paper. Well, I'd like to thank you very much for watching this video, if you are still here, okay? As I said in the beginning, this is my pleasure, this is my hobby. It is something I enjoy to do, okay? I'm studying more HLA, I'm practicing my English. As you can see, I must practice much more, but I hope you enjoy it, okay? And if you enjoy, please leave a comment, give a like to this video. This is a feedback for me that you are enjoying this video. Once more, thank you for watching and see you later. Bye bye.